Hello, good afternoon, everybody. If you are here for the physical therapy session, then you're in the wrong room. But assuming that you want to hear a little bit about barriers for genetic therapies, then you came to the right symposium. So we built this session. Well, actually, let me start with a mandatory disclaimer. So whatever I say, what the speakers say, is not necessarily representing the view of the CF Foundation. We don't sort of curate what people get to say here. So throw the tomatoes at the individual speakers, not at the foundation, please, right? Here, I have nothing to disclose. I just work for the foundation, and my co-chair, Marianne Carlin, also has nothing to disclose other than that she at some point got an honoraria from Vertex and is also in a collaboration agreement with Atlantis. The session itself, David Meyerholz, Bridget Gompert, Anders Wittrup, and Millie Sullivan, we lined up this speaker group because we were looking to take you all through barriers for genetic therapies, basically starting with the sort of course 10,000-foot view at the lung and anatomy that David will take us through. Bridget will kind of take us a level deeper into the specific cells that we find. And then we go inside the cells where Anders will basically tell us how difficult it is and what we can do to get out of the endosome. And Millie at the end will tell us a little bit on how we do get actually out cargo into the nucleus, where many of the genetic therapies, of course, need to end up. With that, I turn it over to Marianne, who will tell us a little bit more before we get going with the speakers.
Yeah, thank you, Martin. So the chairs sort of have different styles of starting the session. And so basically, we try to hopefully recapitulate properly and accurately the research of the four speakers in this line of research, going indeed with increasing granularity from the lung to the cell to inside the nucleus. And so basically, the first speaker, Dr. David Meyerholz, is going to be talking about the lung. And so lung anatomy, I guess you're all very familiar with it. It has many layers of complexity, and schematically, this is what the lung looks like. So we know it has a huge surface area, that there are many different regions of interest. We'll be hearing about these different regions, and that we can also deliver gene therapies from the apical side or through the blood, so systemically. So schematically, this is what it looks like, but ultimately, of course, we're going to be looking at the real lung, and so this is what Dr. Meyerholz will be guiding us through, looking at the real lung and the real cystic fibrosis lung, and regions that are highly affected in cystic fibrosis lung disease, both in humans and in animal models. And then sort of once we understand the structure of the lung, then we're going to dive deeper into the cells that play a role in this disease. And so when we look into the lungs, and in particular the conducting airways are the major region where CF lung pathology and pathophysiology starts in the end, Bridget Gompert will be talking about single cell resolution insights. And so basically, we'll learn more about the different cells in these different regions of the lungs. And so a lot of attention has been put on the surface epithelium, understanding which cells are present in this region and also which cells express CFTR or do not. We'll also hear a bit if it's important to target specific cells for gene therapies, and it might be different from one gene therapy to another. But basically, I think we'll be hearing even more about another structure, which is also something we'll be hearing from the previous speaker, and that's submucosal glands, and diving into single cell insights in that region. And then we're moving to barriers. Well, we'll first start actually with the different delivery vehicles. And we've heard a lot about LNPs, lipid nanoparticles, and the promise. And this has been presented by the plenary speaker yesterday, Dr. Dan Sigurd. And so you probably all recall these images, which are pretty incredible, showing very long-lived and very efficient lung cell targeting, both of the epithelial cells, of the conducting airways, and also of the endothelium. So basically, we can say this is efficient lung cell targeting at this point in a mouse model. But then when we dive deeper into the mechanisms of how LNPs get into the cell and also deliver their cargo, a lot of bottlenecks are uncovered. And so in particular, this endosomal escape is a challenge which really limits bringing the cargo into the cytoplasm and then into the nucleus. And so what the third speaker will be talking about is this endosomal escape, how we can understand it by visualizing it, quantifying it, and in particular also modulating it to improve gene therapy outcomes. And then the last speaker is going to take it all the way up to the nucleus. And so Dr. Millie Sullivan will be talking about different delivery strategies based on peptides and polymers, really for the intracellular delivery of gene therapies. And basically, they go through the same process of uptake, endosomal escape. But actually, what she'll be focusing on, if I'm not mistaken, is this last part of which mechanisms and strategies can be used to really bring the cargo into the nucleus, and in particular, focusing on DNA delivery. So basically, with this, I hope I've introduced, like in a helicopter view, the 4 topics that are going to be covered. And then I welcome Dr. David Merholz to the stage.
And the session runs in a fashion that we actually have 20 minutes talks, a short time for specific questions, and we at the end have all 4 speakers up here for a panel discussion. So anybody who doesn't get to ask that question in between the talks can definitely come back with questions for the panel discussion also.
Do you want me to start the timer? Or just, I don't mind.
No, you can start.
All right. I'd like to thank the organizers of this meeting as well as the Cystic Fibrosis Foundation for inviting me here to speak today. I don't want to speak down to anyone. A lot of you know way more about the lung than I do, but we're just trying to get everybody to a common plane here. There's the disclosure that was shown earlier. My disclosures have no relationships to disclose related to this presentation. I am a little bit animated, so bear with me. I want to provide an overview of the respiratory tract for you today that will give you an understanding, a basic understanding, and then apply that to CF and some of the inherent challenges we have in that. The human lung is really remarkable, and so starting off, I have to do this. Deep breath, hold, then let out slowly. My heart rate has gone down. I'm much less nervous now. If I had done it a little longer, it would have gone down more. But the point is that the lung is an important organ. It's interfacing with the cardiovascular system, with the autonomic nervous system. It's just so fascinating. And in fact, people have looked at the lung, done stereology and that, and come out to the conclusion that every time you're taking a breath, you're ventilating an area about the size of a tennis court. Think about that. That's just really remarkable, isn't it? The respiratory system is composed of several structures. We'll go through each of these, but it has many purposes. First, treat the incoming air. Anybody who's been, well, we have a couple people from Sweden here. I'm sure it gets very cold in mid-February there, and when you breathe in that cold, dry air, it kind of hurts your nose a little bit when it goes in. But importantly, you're humidifying the air, you're also warming the air, so when it gets to the lungs, it's going to be doing pretty well. It also removes particulates. As a kid, I grew up on a farm, and I had to bale hay, and part of baling hay is after you get the hay baled, you got to put it in the barn. The barn has really bad ventilation, and hay's really dusty. Dust everywhere, can't see anything. Anyway, that night when you go to shower, you're like, my nose is full of something, blowing it out, and all this black stuff keeps coming out, and it's all the dust that my nose had collected, keeping it from going into my lungs. So it was doing its job, protecting my lungs. Gas exchange is important as well. Intake oxygen, expel carbon dioxide, and through that process, you can also regulate acid-base balance in your bloodstream. Very important. Detoxify compounds. There's cytochrome P450 enzymes in certain cells in the respiratory tract, and they can protect you from various things, and sometimes they can cause free radical formation that can cause actually lung disease themselves. Neurosensory, smell and taste are important associated with the respiratory tract, and I'm talking to you right now in part because of the respiratory tract. The respiratory tract is generally divided into upper and lower respiratory tracts. We're going to talk a little bit first with the upper respiratory tract, and it's composed of the sinonasal cavity, and then also the pharynx and the larynx area. This is a sagittal view, like straight through my head here, of the sinonasal cavity. And the important part here is showing the epithelium. So this is different colors of epithelium. The blue epithelium is represents the respiratory epithelium. That's pseudostratified columnar ciliated epithelium. That's the same thing that's in a lot of the airways in the lung. So if you're going to treat, say through some sort of inhalation process, treat the lungs, then you may be affecting also the nasal cavity as well if you're targeting certain things like ciliated cells or goblet cells, et cetera. Additionally, you also have here in this little red area, olfactory epithelium. Olfactory epithelium, as we learned from COVID, if you get a virus in there or something, you can actually cause it so you don't smell things properly. Well, again, if you're doing treatments, you want to make sure that be aware that there's a potential for having adverse effects with the olfactory epithelium. The sinonasal cavity, these are little inverted areas of recesses in the bone that form during development. In CF, we found both in humans and then also some animal models that some of the sinuses are actually hypoplastic and may not form well during development. So they're already starting off at a bad start. And then also we have, soon after birth, a lot of kids will have sinus disease, and so they become very full of mucus. And so again, if you're trying to treat those areas, getting in past that mucus is going to be a challenge. Now we're going to go to the lower respiratory tract. And so that starts right about the level of the larynx, where this red line is, and works down. We're going to talk specifically about trachea, bronchi, bronchioles, and the lungs themselves, the alveolar region. In general, there are two functions, so to speak, within the lower respiratory tract. The first is the conduction zone. And anybody who has been on a major tollway going, say, to Chicago, and I say Chicago because we go there a lot, but once you get on, your only purpose in going on this tollway is to get to Chicago. You can't go off and sightsee, it won't let you. You can maybe get gas, but that's about it. Your whole thing is going towards Chicago. And so when you're in the conduction zone here, you're really just getting the air down to the alveolar region and then getting the air back. And so that's the main job. But there is a lot of things along the way that help the air get there. There's secretions and protections like mucociliary transport and such. And the other area is the respiration zone. That's where there's actually alveoli. That's where you can actually respire and have transfer of gases. And that's the respiration zone down in the distal areas, and that's respiratory bronchioles, alveolar ducts, and alveoli. If we were to make a schematic that represented the lungs, this would be a very good one right here. And in general, as I'm looking histologically at a sample, I'll know it's trachea probably by its size. It's a big tube coming down from our larynx. In fact, if you want, you can feel in the crotch of your, right down here, you can feel the rings of the trachea if you push hard enough. Don't make yourself gag or anything. But so they're present, and the gray here represents the different cartilage rings. Associated with cartilage is also submucosal glands. They tend to go hand in hand in a lot of these different tissues. The next one's bronchi, and that's where the trachea bifurcates into two sections going to the different sides of the lung. And we still have cartilage here seen, but it keeps getting smaller and smaller. And eventually down at this point is where it stops. But anyway, these are still called bronchi and associated with cartilage and submucosal glands. The next step is down to bronchioles, and that's where we transition away from the cartilage and away from the submucosal glands and come down just to airways. And there's enough support from the lung structure to hold the airways open, and that's part of the reason we have cartilage there, is to help keep the airways open. And then finally, we come down to the alveoli. Again, this is the site of gas exchange, and this is where a lot of that gas exchange occurs, and really, you got the highway, the conduction system going down to the alveolar region. Now we're going to look at this a little bit more closely. The trachea itself is composed of a large piece of cartilage. It's a big C-shaped piece of cartilage. And one thing you can't really appreciate here, but there's a lot of smooth muscle right in this area. And it attaches to this edge of the cartilage and this edge of the cartilage. Now, when you're watching the World Cup and your favorite team is about ready to score, and they do score, and you have a wad of potato chips in your mouth and you start gagging and one or two pieces go down your trachea, how does it come out? Well, you start to cough. And when you cough, you put pressure on air going out. And then your trachealis muscle says, we need more force, and it pulls the C-shaped cartilage together to make the airway narrower and accelerates the force of the material going out. That's how we help to get stuff out of our trachea. This is an example of tracheal histology, and one of the things you can note here is it's got cilia it's got these little pale areas, which is some mucus change going on in those cells. We also have pseudo-stratified, meaning the nuclei look like they're piling on each other, but they're really not. It's a fake image here, but it's pseudo-stratified, meaning it goes all the way from the top all the way down to the basement membrane. And so anyway, this is the classic appearance of respiratory epithelium, similar to what we see in the nose to some extent. Now, I would normally ask this in a question, but since we're on a limited time here, I'm not going to do this, but this is a little blood vessel here. This is most likely at the capillary level. And one thing that is interesting, if I went and wanted to have gene therapy and put in a catheter right here and injected it into my body, surprisingly, this capillary bed is not the first capillary bed that's going to see. Which one is it? Where's the first capillary bed? If I inject into my vein here, where's it going to go? Capillary bed is going to be the lungs. So the peripheral, the alveolar region, we'll talk about that in a second. But it has to go out through systemic circulation again, come back through the bronchial arteries, and then come back here to the airways. The airways are where we're treating. So you need to think through this because you may lose some of your substrate if you're injecting peripherally, or you may have some off-target effects down in the distal lung if you do it peripherally in a systemic way. So those are some considerations as well. And again, here's the surface epithelium, some lamina propria, which is connective tissue, and here's some submucosal glands. A bronchus is farther down into the airway. And again, this is a different stain, but again, you have these bubbles like here. This is all cartilage. These colors right here are actually representing submucosal glands, and this is just a collapsed airway, possibly from just postmortem change. And again, we have a pseudostratified ciliated columnar epithelium. As we get smaller and smaller, the colors don't change, it's just a different stain, so don't let that bother you. But you can see the airway surface epithelium is a little bit thinner. You still have some cartilage here, these little bubble areas. You also have a little bit of submucosal glands, but this is a small bronchus. Now on this side, you still have a thin area of epithelium here, relatively thin, but you don't have any cartilage, you don't have any submucosal glands, so this is actually a bronchiole. At the very end of the bronchiolar region is the terminal bronchiole, and that's an area where it dumps out and starts getting into the respiration zone. And usually in humans, we'll see it in the next image, but usually it goes into a respiratory bronchiole, which is half bronchiole, half alveolus lined. But anyway, the point is that it's the end of the conduction system, and that conduction system is reaching the alveoli. One other point here at this level, notice that the nuclei no longer seem to pile on each other. This is almost a simple columnar, simple cuboidal area with cilia still. Not as much cilia, because now you're getting down to the area where you're starting to get more club cells. Now, when we look at the alveolar region, we can piece all this together with this one nice image. We have a terminal bronchiole. It goes right into a respiratory bronchiole, which has partially lined by alveoli, as well as some wall that looks like a normal bronchiole. You get down here to alveolar ducts, which is just a passageway that's lined by alveoli, and then finally down to the actual alveoli down here. So at the alveolar level, we have a bunch of different cells down here, and these look like a bunch of eggs that are being cooked on the skillet, doesn't it? And it's not so much that they have a raised yolk area here, that's just the nucleus, but these are simple squamous cells that are very, very thin. It has to be thin because we have to get air exchange occurring down in the alveolus. Additionally, we have type 2 cells, which are the green ones here represented. This is the one that produces the S surfactant that allows the airways to be able to, allow the alveoli to stretch and reduce the surface tension. And we also have some alveolar macrophages in here as well. Importantly, as I mentioned before, if you gave a peripheral injection, this is the capillary bed that's going to be first seen by your injection down here in the alveolus region. And all these capillaries are where that's going to go. This is just a transmission electron micrograph, and this shows how small it is. This big structure right here, this is not a huge island or anything. This is an erythrocyte, an RBC, and that's how close we are in magnification. And this is a basement membrane for the endothelial cell as well as a type 1 pneumocyte. So again, this is a very small area, very thin area. And if you have a bad viral infection and you get interstitial pneumonia, this area gets thicker and it makes it harder for gas exchange. And that's why you may sometimes have to go to the hospital and get some oxygen to supplement. In general, the caliber, the width of the airways gets smaller as you go down. The epithelial height goes down as you go down the airway conduction system. The glands go through the trachea and bronchi and generally get less, and the cartilage gets less, but these are not present in the bronchioles. So now I'm going to get to the challenges associated with genetic therapy as I think about it. First is airway obstruction, and this is an airway from a 2 to 3 month old baby, circa about 1945, World War II. And this baby has airways full of neutrophils and infection. This is a lung transplant from a patient probably in their 20s, and again, we have lots of neutrophils and some mucus in this. So airway obstruction is going to prevent access to airways and epithelium. So that's going to be something that's inherent in CF, and it starts at an early age. In Anderson's first report of CF in 1938, 49 autopsy cases, 44 of them were determined to have died because of airway infection and inflammation. So it starts early. Another challenge is airway geometry, and some of you may be wondering what that means. It's not a math course, but it's where there's different shapes of the airways have been made in a way that's not normal. And sometimes they're smaller, sometimes they're shaped wrong. And this population in which a lot of these were studied is young pediatric cases. Now, I would argue, we don't have proof of this, but there's a lot of substantial evidence saying that this population that has these airway geometry problems are also the same population that's going to need gene therapy. They're going to have severe mutations of CFTR. And so one of the things to think about here is, how does airway geometry, which probably happened in fetal life, so it's going to be there as soon as birth, how is that going to influence how we give different therapies to these people? It's going to influence airway resistance, particulate deposition, and even where the aerosols go. Another issue is that of remodeling. And when I say airway remodeling, I tend to think about the epithelium itself. This again is pseudostratified epithelium with a little space up here. So that's kind of normal. Over here we have an area that has nuclei going all the way to the surface. Additionally, we have a little bit of increased fibrosis here from this paper, not as much here. So again, you have some changes going on that's going to maybe limit the access to some areas. It's also going to maybe have some changes that's going to make it harder to have the right cell type there. So the cell types may change. We've heard some talks already here at this meeting where ionocytes are different in different parts of the lung and the airways, and that sometimes CF versus wild type's different too in numbers and even phenotype of those cells. So those are issues that would be important to know. And lastly, just as on this section here, just talking about as these airways get filled with this pus in there, it loses the ability to get good vascular supply there. It's called venous infarction. And what ends up happening is, is that the airways will actually rupture, and that's where you get an abscess formation in the lung. So again, changes to cellular access, target numbers, or target receptor expression may be limited. One area I'm very interested in is the non-epithelial changes in the lung, and what does that mean? Well, this represents smooth muscle around here. CFTR is actually a smooth muscle relaxant when it's activated. Neural cells, so these are like nerves and ganglia. They play a role in reflexes that occur in the lung. We also have immune cells that are sitting out there, and we know from other talks here that immune cells are sometimes affected by CFTR loss and can actually have a hyper-inflammatory phenotype and such. And then just these neuroendocrine bodies here, the neuroendocrine cells. So if we're just putting in a topical treatment and we're not thinking about these other cells, we may help improve the CF aspect of the patient, but at the same time, we may also go and make a more enhanced asthma phenotype in the patient. I don't know, that's just a guess, but I'm sure people have ideas about that. So uncorrected non-epithelial cells could cause persistent lung dysfunction. Lastly, as we think about the cell type and where that's located, I think about a lot of different cells. One easy way of thinking about it is in the submucosal glands. Submucosal glands have these far off distant areas called serous cells, and they have nice brown staining here, meaning they have a lot of CFTR. The mucous tubules don't have as much, and if you get out to the ducts, they have a bunch. I analogize this to being at a water park. Has anyone gone to a water park? I know you all have. But if you're at the top, you want to have a lot of water to get you going down the slide. Once you're going, the thing you don't want to do, which I've had happen to me, is get stuck at the bottom and not make it all the way. So you want to have extra water down at the bottom to get it out. And that's kind of what happens with the submucosal glands. You want to, it's targeted appropriately to get the mucus out of the submucosal glands. But if you do gene therapy of, say, a progenitor cell for the submucosal glands, what happens if you have CFTR expression enhanced in the mucinous tubules? How is that going to affect? Is that going to perhaps enhance liquid secretion and cause problems? So anyway, those are things that I think are important to think about. And in general, paralleling the native expression levels of CFTR in cells would seem advantageous, which also follows structure-function relationship, which as a pathologist, I like to see all the time in just nature alone. So anyway, I think that's something just to think about. And I want to thank everybody who's helped me ever in my life. I know this is an incomplete list, so that covers it. But anyway, thank you for your time.
That was a great first talk in the session. We have time for a couple of questions. That's why people think about that. I may ask you one. So you pointed out that there's actually two different circulation circuits into the lung, and we really don't want to be in the pulmonary artery, but rather in the bronchial one. So can you think of strategies on how you get directly to the bronchial artery?
Without having a lot of invasiveness, I've been thinking about that too, and I can't come up with an idea yet, but when I do, I'll sure let you know.
Do you think catheterization to go there directly would?
I think it's definitely a possibility, very similar to what they do with, when they do the heart work, they can go up through the inguinal vein or some other places that maybe would get it to a better spot, but you really have to go backwards in the system and start from the periphery of the arterial system to get back to where you really need to go. And so I think it would be a challenge for sure, but I think there's ways possibly to get around that through affinity markers or something that would bypass that first capillary bed, or something you give ahead of time that would bypass that first capillary bed and allow it to go to the second one. Sorry.
Along those lines, I mean, the submucosal glands, they have the same capillary bed as the right under the subepithelial surface.
Yeah, so those would be supplemented in the wall there. So if you think about it down in the pulmonary area, the alveolar region, it's got oxygen and gas exchange all at once because it's down there. But if you get up in the wall of the airways, they don't have that kind of luxury, so they have to have their own blood supply, and that's where it comes from, the bronchial artery. So the submucosal glands would be included in that.
Caroline.
Thanks. I was wondering if you could comment on animal models for CF, right, and how we can think about drug development in relation to the animal model physiology and the human respiratory physiology.
Oh, okay. It's a little bit more than I was thinking about for this talk, but yeah. So I think having some sort of relevant biomarker or some sort of relevant feature that you're looking to test. So let's just pretend we have a very severe phenotype in a human as far as a mutation for CFTR. They're going to be the ones that are going to be the most likely to need gene therapy or genetic therapy. If you were to take some of the null animal models that have been made, they actually could be useful to look at to see on-target effects as well as off-target effects. And when some of those first models came out, they were like, that's not quite CF because it's too severe. But in reality, it's mimicking the severe phenotype that's seen in that small, 10+% that we see in the human population too. So I think it's going to be a useful opportunity for some of those models to lead the way in gene therapy testing.
Yeah, so I have one more question. So I was just wondering about the theory of the distal airways, that CF disease would start in the distal airways before really moving up towards the more proximal airways. Is this something you think we should be targeting like early disease and that it's also a different part of the lungs?
That's a good question because like even if you think about ionocytes, sometimes they have a different phenotype depending on if they're lower, mid-level, or higher, and then also they may have different co-transporters associated with them. And so is that really a phenotypically the same type of cell that you're treating down low as it is up high? Is it going to be absorption or is it going to be secretion? There's a lot of issues there, and I think even the ionocyte session yesterday was really good, and it talked about a lot of those issues that we need to be thinking about, about what type of cell we really want to target, and if that cell is in the right location to be appropriately targeted.
Thank you, David. We will have David up here again if people have questions later for the panel.
Well, I'd like to thank Marianne and Martin for this opportunity to speak here today. And I've been charged with talking about targeting and particularly thinking about which stem cells, which stem and progenitor cells should be targeted in the airway. And this is giving me the perfect opportunity to share with you some very new and unpublished data on submucosal glands. And in particular, I want to talk about the submucosal gland stem and progenitor cells. So I'm going to just continue on the anatomy theme just for a little bit longer, though I'm really grateful to David for that beautiful introduction to submucosal glands. But I did want to highlight some structural issues within the submucosal glands that I think are important to realize. And that's that when you start at this is the most lateral in the submucosal glands, we talk about medial to lateral. There are these serous acini, and they cap these mucus tubules, and these mucus tubules are quite highly branched. And this is actually quite unique to humans in that we don't really see this situation in ferrets and pigs, which is actually really interesting. And these mucus tubules all arrive in this collecting duct, which is quite extensive in humans, again, different to ferrets and pigs. And this collecting duct, let me go back. This collecting duct then leads into the ciliated duct, which through a virtual space then allows these sheets of mucus to come from this mucus tubules through the collecting duct, up through the ciliated duct and onto the airway surface. And so we've been really interested in trying to define what are all the different populations that are present in these different regions of the submucosal glands. And so to do that, what we've done in the lab is to take human samples that come to us. Most of the samples that we get are from patients who are lung donors, and we're able to get part of their cartilaginous airways. And some of these are older patients who are research donors and are not considered good enough for donations, but we get a lot of tissue in. And we take this intercartilaginous zone, which is the region between the C-shaped cartilaginous rings that David talked about earlier, and we actually excise the intercartilaginous zone. And that's because it's a region that's very rich in submucosal glands. And the first thing that we do with the intercartilaginous zone is we want to make sure that when we're profiling the cells, we're not profiling the surface airway epithelium at all. So we actually mechanically strip off the surface airway epithelium. And this is an H&E slide showing you that this is the basement membrane over here, and there's no surface airway epithelium. So everything that I'm showing you in terms of profiling is below the surface airway epithelium. And then we go ahead and do an enzymatic digestion and isolate the glands from there. And so this is the cell subtypes that come out of that intercartilaginous zone. And it's really striking for how many cell types there are. And I think it's not surprising that there is a large number of immune cells, which are shown on the side here. A large number of endothelial type cells, including lymphatics. And there's some mesenchymal stromal cells as shown over here. But what I really want to take a deeper dive into today is actually looking at more of that epithelial submucosal gland compartment itself. And I also want to point out that all of this work has been done by Andrew Lund, who's a very talented graduate student in my lab, together with Justin Langerman, who's a fantastic computational biologist from Catherine Plath's lab. And so this is the populations that we get. And the way I'm thinking about them right now, and of course, this may change as we dive more into the data, but we've been sitting with the data for a number of years now, already trying to think about what it all means. And the way I'm thinking about it right now is that there's two major cell compartments. There is this compartment here up at the top, which is really related to the ducts. And there are duct basal cells and cycling duct basal cells that to me are very reminiscent of what we saw in the surface airway epithelium with the surface airway basal cells. And then there are transitioning cells, again, very similar to what we'd seen in the surface airway epithelium, this time with duct cells transitioning to secretory cells and club cells, some duct basal cells that are becoming what we're calling duct luminal cells, because we think that these might be part of collecting ducts. And then there are these ciliary populations that always cluster very uniquely and separately. And then, in my mind, even more interesting is this other cell compartment down here, which is really very tightly associated with the myoepithelial cells, which are the cells that line the tubular acinar structures and are really critical in contracting to move the mucus down through those tubules and into the collecting ducts. And these myoepithelial cells have such a strong muscle signature that they actually pull in the smooth muscle cells and the myofibroblasts into what we would otherwise consider an epithelial plot. And this is, again, really interesting because it highlights that there are several populations that are transitioning. So I think one of the themes that we've taken away from the single-cell sequencing is that these cells are very dynamic. You're not just sitting in a progenitor state and a differentiated state. There are many, many different transitions in between, which we think are really interesting. This may also be because, as we know, humans are constantly exposed to environment and infections and may have diseases, and so there might be a lot of turnover of these cells. But what we're seeing is these myoepithelial cells and these acinar transitioning cells leading to this very interesting tubular acinar progenitor. And this progenitor is really a secretory cell progenitor because it can become a serous cell, and so through a serous progenitor to a serous cell, or through a mucus progenitor to a mucus tubule cell. So it's really poised to go in either direction in terms of secretion. And then there's also another progenitor cell that we had not expected to find, and this is what we're calling a myomucosal progenitor. It has strong muscle signatures, but it also has oglycosylation and more of a mucus cell signature, so it has both. So based on this, we started to think about, well, which cell gives rise to which cell, and can we predict, even though we can't do lineage tracing, or it's very difficult to do lineage tracing in human tissue, can we predict which cell is giving rise to which cell? And basically, what we've been able to do now with monocle pseudotime analysis is to try to identify, to say that we think it's the myoepithelial cells that go through this acinar transitioning cell and then into tubular acinar progenitors. And then from there, the tubular acinar progenitor does its serous and mucus thing. And so that's the way we think that the myoepithelial cells are working towards the tubular acinar bipotential progenitor. But what's also interesting is that it seems like the myoepithelial cells can also transition through this myomucosal population to become straight away into a mucus tubule cell, and then from there go through potentially into the tubular acinar progenitor. And so it seems like there are actually two routes by which you could make a mucus cell for the mucus tubules. And this is highlighted also over here, where we're showing the myoepithelial signal going through this transient and then transitioning acinar population through to the bipotential progenitor, the secretory potential. And then here is that myomucosal progenitor, which seems to be able to go straight into the mucus tubule cell, differentiated cell. So we've also been thinking a lot about, so how does the submucosal glands, the different populations in the submucosal glands, how are those related to the surface airway epithelial cell populations that we published now several years ago? And so we've overlaid the two signatures, and I think some of it is not surprising, but I'll show it anyway. So we found 3 different basal cell populations that overlay onto the submucosal glands. The canonical basal, which seems to be in the positions that we would expect, cycling basal, as well as the basal to secretory transitioning cells, which seem to go through the serous progenitor, which I think is not surprising. We also have, obviously, really beautiful overlap with cilia, and cilia is oftentimes just the most obvious signature that we get. And then we also are able to see the secretory transition to cilia, which interestingly enough is actually coming through more strongly with our mucus tubule signature. And then we have, sort of as a reality check for us, this is our control. When we actually did our surface airway epithelial analysis, we included in there some submucosal gland samples. And when we went back and looked, our secretory 5 population was actually uniquely lactoferrin lysozyme producing cells, serous cells, just uniquely to the submucosal glands. And so we see that again overlapping very nicely with the serous data in our submucosal glands, showing that this indeed is exactly what we had found and was not unique to surface airway epithelium. So for this audience, I'm sure everybody wants to know, so where is CFTR expressed? And so I almost showed this slide with a bit of pause because this is not where we were expecting CFTR to be expressed. But this is where we find it. We find it most strongly in the tubular acinar progenitors over here. And it's almost like it starts to come on as the myoepithelial cells go through these transitioning acinar cells. And it's there in the mucous progenitors too, and less so in the actual mucous tubules. And then we do see some in the serous cells and within the duct and some of the transitioning duct cells. But it's really this tubular acinar progenitor population that we were really struck by. And I also want to emphasize that this is gene expression, and we're still working with Jed Mahoney at the CFFT to look at the protein expression, which I think will be very interesting. But because of this, and because we really needed to validate the single-cell RNA sequencing, Andrew Lund decided that he really needed to create an organoid model of submucosal glands. And so what he actually does is he takes those digested submucosal glands, places them into Matrigel, and has figured out a number of growth factors and a whole cocktail of things that are needed to take them through what he calls an expansion or patterning phase. And he does about 10 days of this. And then there's a different cocktail, which allows differentiation to happen for the organoids. And what I'm showing you here at the bottom are some immunofluorescent images just showing you what these submucosal gland organoids are producing. And in the expansion phase, this is the keratin 14 staining, showing you that we're just getting a lot of expansion of the stem and progenitor cells that are characterized by keratin 5, keratin 14. And in this expansion phase, we don't see any MUC5B expression or lysozyme expression. But after differentiation, now we start to see some MUC5B turning on and some lysozyme turning on. So we were very excited about this, and we got in touch with Jed at the CFFT, and he and Alex were wonderful about saying, okay, well, is there functional CFTR in these organoids? And he got us back some data that we think is very interesting and really sparked further experiments in the lab. And that's because with forskolin swelling, we see some cells, as marked with the white arrows, that do swell at this 4-hour time point. But as marked with the red arrows, we see some of the organoids that don't swell. And of course, some of this can be inhibited with the CFTR inhibitor, and it's quantified here in the graph on the left. But what I think it's telling us is that the submucosal gland organoids that we're making are actually quite heterogeneous. And that, going back to the single-cell data, makes sense because we probably have some duct stem and progenitor cells and some myoepithelial cells, stem and progenitor cells. And so there probably is a lot of heterogeneity within our cultures. And we were also really gratified to see that during the expansion phase, we don't have CFTR expression. But with differentiation, this is 14 days of differentiation, we do start to see CFTR expression coming on, and this is just a primary enteroid as a control. So this really made us decide that we had to figure out some markers that differentiated the duct stem cells from the myoepithelial stem cells so we could grow different organoids. And so this was actually not that easy to do, and that's because as we were doing more and more immunofluorescent staining and really following the surface airway epithelial basal cells down into the glands, we realized that there's really a stem cell and a basal layer continuum throughout the airways. And we're using alpha smooth muscle actin here as a marker of the myoepithelial cells and some of the myofibroblasts, but just to orient you, and keratin 17 is this beautiful basal marker that goes all the way from the basal cells on the surface airway epithelium, dives down into the ducts, sorry, this arrow is a little tricky, sorry, and then becomes flatter and thinner the further out you go down the ducts, and eventually aligns and forms the myoepithelial cells. So we figured that there wouldn't be probably anything that was a specific marker for a duct progenitor, like a basal duct progenitor cell versus a myoepithelial cell. And so what we did is we went back to the single-cell RNA sequencing, and we said, okay, so can we identify some genes that look like they might be good candidates based on having high expression in the duct and low expression in the tubular acinar cells, and vice versa. And so we did some immunostaining to validate some of those, and we did, as I'm showing you here on the left, we did find some markers that were higher in duct than in the tubular acinar structures, as here, and we also found some that were more highly expressed in the tubular acinar than in the duct. And so we developed a FACS sorting strategy. And this is our current strategy. And we're actually working on newer markers now because we've done a lot more work. And we think we have better markers. But this is the best data that I have right now. So I'm going to show it to you. So essentially what we do is we do quite a permissive gate on NGFR and EGFR, as shown over here. And this really is just enriching for the progenitors. But there's a lot of other cell types in here, too. And then we use integrin alpha-2 to separate out these two different populations. And if in gate P6 and P7 here, if we take those cells and we culture them, they really just grow like mesenchymal cells, like stromal cells. And they don't resemble epithelial cells at all. But the P8 and P9 populations are very interesting. And when we sort those and then grow them in that same organoid expansion and differentiation phase, this is what we see. So this is the parent population, and these were started from unsorted cells. And I think you can appreciate that there is heterogeneity. There are these very large organoids and these smaller organoids. And now if we look at P9, which is this population over here, which we are thinking of as the submucosal gland duct basal population, these are pretty small organoids as compared to P8, this population over here, which gives these very large, very juicy looking organoids. And we think that those are derived from the myoepithelial cells. And to compare, we also took the trachea, the donor-matched tracheas, isolated the surface airway epithelium, digested those cells, and used NGFR and TROP2 to sort for the basal cells. And those organoids actually look pretty similar to the duct organoids. So we thought that this was really interesting. And so we decided to do some more single-cell RNA sequencing on these different organoids. And the data is very fresh as of like last week. So I'm just going to show you some of our initial analysis, but it's looking really interesting. And what we do find is that the duct organoids separate quite nicely and cluster quite differently from the myoepithelial cell organoids. And we do have some secretory populations, and the MUC5B tends to be quite close to the lysozyme lactoferrin expressing cells up here at the top. But we were also really interested to see that there's a lot of populations, even within our myoepithelial cell organoids, that we thought should be there making these very differentiated, mucus and serous secretions that are not. And also what we found is that we did see CFTR, but it was mostly in that tubular acinar progenitor. And so our reasoning was, well, we seem to be getting some differentiation of these organoids, but it's not great. We seem to have a lot of cells that are stuck in a progenitor phase. And so this happens a lot with pluripotent stem cell cultures, as well as with just, primary cell cultures. And so we wondered what growth factors or what kinds of niche factors might be needed to really push differentiation. And so this took us back to our single-cell RNA sequencing, and we did some receptor ligand analysis that I don't have time to show you, but basically what we found is that the ERBB factors had what looked like a very important role, especially in terms of interactions between the epithelium and the mesenchyme. And so we did some immunostaining for some of these factors, and we found, for example, that heparin-binding EGF really localizes a lot to the myoepithelial cells and some of the surrounding mesenchymal cells. NRG1 is really in the mucous tubules very nicely, as well as in some of the surrounding mesenchymal cells. Epiregulin, very much so in the surrounding mesenchymal cells, as well as some of the serous cells. And EGF, a lot in the serous cells and some of the surrounding mesenchymal cells. And so we started to think about, we wondered, could some of these ERBB ligands do some differentiation or change the potential of these cultures? And so we ran a mini screen, and we came out with some interesting results. And from this mini screen, we found that if during the expansion phase, we added particularly the combination of HBEGF and NRG1, we could take our organoids, and these are the parental organoids, unsorted organoids, and really expand the branching and the budding. So we could make them look a lot more like submucosal glands. And in our minds, we're reasoning this could be something like a submucosal gland hypertrophy, hyperplasia, potentially. Then we said, okay, well, if that's true, then we should be able to inhibit the ERBB factors, and we should be able to shut down the organoid growth. And in fact, that's true. If we hit it with a big gun like an EGFR inhibitor, we do not get much expansion. And even with a more targeted ERBB3 inhibitor, we shut down expansion too. So it seems like these growth factors are very important in that expansion of the submucosal glands. And so what about differentiation? Well, it turns out we can actually push differentiation of the organoids either towards a mucous cell fate or a serous cell fate. And EGF seems to be the big player in this. So EGF very beautifully will increase the number of mucus, particularly MUC5B expressing cells within the organoids. And Epiregulin on its own, but in particular in combination with HBEGF, increases both the mucous cells, but much more so the lysozyme expressing cells. And so we're very excited now that we think that therapeutically we could actually potentially think about just submucosal gland hypertrophy and hyperplasia and how we could perturb that. And potentially we could have less mucus on the surface, which might reduce the barrier to gene therapy in that way. And in addition, we think that we have other additional targets that might be good to think about for gene therapy. So in summary, what I've shown you today is that we're really thinking about this as two main cell compartments, a myoepithelial cell compartment and a duct stem cell compartment, but they are a continuum, and exactly how they're related, we still need to figure out, and we very badly need lineage tracing studies for that. We are able to generate these submucosal gland 3D organoids, and we can get them to make MUC5B and lysozyme and CFTR and functional CFTR. We have found that we can find different markers that allow us to enrich for populations that are more from the duct or more from the myoepithelial cell compartment, so we can grow different kinds of organoids and assess that biology. And in addition, we think that the ERBB factors are actually really important in the development and differentiation and expansion of these submucosal glands, and that this potentially could be an avenue for the development of therapeutics. So I already acknowledged Andrew, who's done such an amazing job with this, and Justin, who's just incredibly amazing with the computation. And again, I'd like to thank Jed and Alex, and I'm happy to take any questions.
A very nice talk. I was wondering about your organoids, another approach at looking at those organoids, if they appear heterogeneous, so to speak. Have you tried immunofluorescence to try to preemptively identify those and then apply different treatments to those to see how much different cell populations expand? And you might be able to just learn some really new really intriguing stuff that way.
Yes, that's a fantastic point. We haven't done anything like that, but I think it's a great approach. We're thinking about putting reporters in, playing with all different kinds of things, and then probably we can improve our sorting strategy, I think, as well, to make them even more pure. So that's kind of where we're going. Thanks.
Tom Lynch at Nationwide Children's Hospital. I mean, wow, that was fantastic. I can't wait to cite your paper.
That's what we can accept it.
So two questions. One is, it seems like, correct me if I'm wrong, but it seemed like your strategy is mainly to get to a single cell suspension before you do your organoids. And I'm curious, I mean, the alternative is oftentimes people will try to not necessarily get to a single cell suspension for organoids, and how that might affect the structure that the organoids kind of form. And then the second question, if you'll entertain it, is how specific is some of this differential biology signaling pathways to human, and could you tease out some of the, can you better resolve some of the kind of mechanisms using mouse models? You specifically using your organoid system.
Okay, so I can start out by saying that when we started doing the organoids, we did not get to single cell. And so we were using small clusters of cells. So we basically digest out the submucosal glands, and we can actually freeze those down, which is very helpful for us, when you're getting a long day of transplant tissue coming in. And then we can thaw those, digest those a little bit further, but they're still clumps of cells, and then put them into matrigel. With the sorting strategy, we had to get to single cell, that works. So I think it's because we have such a cocktail of factors in the media. And so I think that we're able to bypass not having true feeders or any really true mesenchymal cells with the sorted cells in that situation. But I do think we're doing different things when we grow those two different kinds of organoids. So does that answer your first question? Great. And then the second question in terms of how does human apply to mouse and what can we learn from that? Is that the question? So I'm a little bit wary of the mouse because mucus is just not a driver in mouse. And again, we keep them in these very pristine, mouse houses where the air is so good compared to what we breathe in. And so I think the turnover is very low. And my biggest concern is that I think the turnover of basal cells in the human surface airway epithelium is much higher than it is in the mouse. And I think My personal thought is that the ducts and or maybe even deeper down into some of those mucous tubules and or collecting ducts might be the true sources of the cells that are arriving on our surface airway epithelium. And so what I've been really worried about in terms of gene therapy is if we can get to basal cells, will we be long-lived enough? And I don't know the answer to that because it's so difficult to do those kinds of studies. But that's the kind of things I've been thinking in that regard.
Hi, Bridget. Nice work. I might have missed it, but did you do any single-cell analysis on a mixed population of organoids to see, and then do some cell chat analysis to see if there's any cell-cell communication? Because some of the pathways that you want to look for differentiation may be through crosstalk from the two different organoids. I might have broken this, sorry.
No, that's been difficult earlier today.
Yes, so that's a great question. So we did, and all of that data, we have so much data from all of the organoids we're looking at. We haven't done the cell chat yet on the organoids, so we don't really know what the communication looks like. But all of those things are in the works, and we're really excited to see all of that. Thank you. It's a great question.
In the interest of time, we move on, but thank you, Bridget, and we will have you back up here also.
So then we're happy to welcome the third speaker, Dr. Anders Wittrup, for his talk about endosomal escape.
Yes, I want to thank also the organizers and the foundation for the opportunity given to me to speak here today, especially since I'm slightly out of field, being a cancer doctor, oncologist, neuro-oncologist in Lund, Sweden. And in Sweden, we are more collectivist than you individualistic Americans. So we even have a saying for the horror of being the odd one out, and that's being the cat among ferrets, which seems appropriate at CF meeting. So today, now you're going to hear the cat guy talk about how to overcome the endosomal escape barrier for genetic medicine. So I haven't been in CF research, but I've been into delivery challenges, research into delivery challenges, and especially the endosomal escape step for a long time. And I have nothing to disclose. And just to recap a little bit about what kind of genetic medicines we're trying to deliver. It's a wide difference of different compounds. It ranges from small antisense oligonucleotides to double-stranded siRNAs, mRNAs, or even larger CRISPR compounds. And common for all of these compounds is that they are large, comparably, and charged molecules with very limited bioavailability into the cytosol of cells. So while it's relatively achievable to have these molecules to be endocytosed into endosomes, in most cases only a minute fraction of the cargo that you try to deliver gets out of the endosome, the so-called endosomal escape step, and this is really generally very inefficient. Still, we have developed a number of different delivery strategies, or delivery vehicles that can actually overcome to some extent this barrier. So you've all heard about, and most of you have received yourselves, compounds based on the LNPs, lipid nanoparticles that can be used to deliver both smaller and larger nucleic acid cargos. Also advanced into the clinic is so-called GalNAc conjugated siRNAs. So that's a carbohydrate-based ligand that you can attach to siRNAs and use that for receptor-mediated delivery. And also advancing now into the clinic are various forms of lipid conjugated RNAs that look very promising for local delivery in some cases. So we can use these compounds to get some degree of delivery to the liver, especially. However, to reach tissues beyond the liver, to extrahepatic tissues, we really need to improve the efficiency of our delivery vehicles. And the most obvious target to try and improve is the most inefficient step, namely the endosomal escape step. However, the endosomal escape step is really perhaps not that total black box to our knowledge, but perhaps rather a gray box that we don't understand very much about. So what we know is that we can supply an extracellular RNA therapeutic in some form. We can get the biological effect, but exactly how you get this through this inefficient step is really ill understood. So that's the focus of the work in my lab. And the questions we are asking ourselves are, of course what's the mechanism of this endosomal escape step? But we're also wondering, where does it happen? Where in the cell? When does it happen? How much is delivered? What's the actual efficiency of the step? Are there cellular factors that can facilitate or inhibit the process? And how can we, based on the knowledge we can gain, how can we devise new strategies to improve it? So this will be the focus, especially on our strategies to understand this process, and this will be focused on my talk here. So I came into this field now quite a few years ago when very, very little was understood about the endosomal escape. And then the most simple model system that we wanted to use to try and visualize and understand the process was transfection lipid-mediated RNA delivery, or in this case, siRNA. And transfection lipids, then you get large particles with lipids that somehow you can get it into the cytosol cells. And here you have in magenta labeled RNAs. And if we play this movie, you can see the cells, they chew up these particles, but it's not obvious that you get anything out into the cytosol. And this was pretty much the state of the field. So then we devised a couple of different imaging strategies to really pick up the faint signal of fluorescent siRNAs inside the cytosol. And then it looks like this instead. So then we can visualize the sudden filling up of the cytosol of cells with a fluorescent RNA cargo. And then we actually see the redistribution inside the cytosol afterwards as well. And in this process, we also stumbled upon another family of proteins, which are now my favorite family of proteins, the galectins. And those are a family of proteins that are involved in patrolling the integrity of the endosomal system. So these were discovered or shown to have an activity in this kind of activity for when bacteria damage endosomes to try and get into the cytosol of cells. And then these galectins, they are present in the cytosol, and when the endosome is damaged, you have an exposure of a complex carbohydrate structures on the inside of the endosome, and then the galectins are recruited and mark this vesicle as damaged to the cell. And we especially identified one member, galectin 9, which is very rapidly and very potently recruited to any damaged endosome. So we can use these galectins for various purposes. So here we have, once again, cells that are incubated with lipoplexes, larger particles. And you will see here on the right-hand side, you see the RNA, and you can see the endosomal escape. And on the left-hand side, you will see the galectins being recruited to individual endosomes that are damaged. So here we have a vesicle, and at the very same time as we have a very weak signal in the cytosol, we have recruitment of the galectins. And then we have another event here with distribution of the RNA into the cytosol, and at the very same time, we have recruitment of galectin. And here we have two more events, more clearer, where we have a cytosolic distribution. And then we can also use the intensity of the RNA cargo in the cytosol to estimate the number of molecules that actually leaked out at every event. And then we can use that information to probe compare it to the biological activity and get a kind of a dose response in a cytosolic RNA delivery. So here are individual or grouped cells that have had various degrees of release into the cytosol, and then we can see what was the biological activity in those cells. We can use also these galectins as sensors to screen molecules, small molecules, for membrane damaging activity. So here are two compounds on the right that over time, when we incubate cells, trigger widespread membrane damages seen as these spots of, in the intensity of the galectin. We can look in more detail to see, okay, if we co-incubate cells with ligand conjugated RNAs that are taken up by cells, then they end up in these endosomes, as you see. But then we also add a small molecule drug that induces damage to the endosomal system. What does it then look like? So then we have, for example, this vesicle here that suddenly releases the integrity of the endosomal membrane is affected, and we have a sudden, I'll show it again, release of the cargo inside, and at the very same time we have recruitment of this galectin sensor. But as you can see, it's still very inefficient. Most of the cargo is inside the endosomes. We can also use the galectins to screen for activity in other kinds of systems. In this case, it's a tumor spheroid and organoids we can use. And then here we've also used small molecule drugs to induce these damages. And then you can see the spotty appearance of the galectins as evidence of membrane damage. Another class of molecules that we have been interested in as a potential enhancer of endosomal damage and endosomal release are lipophilic compounds in a family called triterpenoids and related saponins. And we've screened various members of these compound classes and identified especially one, ursolic acid, that seems to induce very widespread damages with very low toxicity. And if you combine that with an siRNA targeting GFP, you can go from very limited knockdown to very potent knockdown. And still you have to use this at quite high doses despite low toxicity. So it's probably not the perfect drug, but you can see what it looks like a little bit. So here we have a cholesterol RNA in magenta, and to the right we add ursolic acid, and to the left it's the control. And you can see in green very rapid induction of very widespread membrane damages, and still the cells are surprisingly basically very healthy. And what we also can see is that we get much less accumulation of intracellular structures with the non-released cargo. Since we can then use these galectins to probe the mechanism, we were very interested in using them to understand better the delivery using the very widespread used LNPs. So we've studied LNPs with both mRNA and siRNA using these tools, and in both cases we can see that if we pour these on LNPs on cells, we get widespread recruitment of galectin on the LNP containing endosomes, but not on all LNPs, but on a subset of them. If we look using live microscopy at an individual LNP or individual endosome, for which we have recruitment of the galectin, we can see at the very same time as we have recruitment, we can observe a drop in the cargo inside the vesicle. However, this is a single example. If we look at large numbers of vesicles, what is quite apparent is that the intensity of the cargo inside the vesicle, that there is a drop at the time of recruitment of the galectin damage sensor, but it's quite limited, so it's quite inefficient. Irrespective actually of dose of the RNA. What we could observe was that we could divide the events with recruitment of galectin in two groups. Either there was basically no release at all despite damage of the endosomal membrane, or there was a group in which we had efficient, rapid release, but still not complete release. And the ratio of these, if you had release or not release, was around 50% of the cases there was actually release. And if there was a release, around 50% or so could be released. With mRNA, it was actually worse. So only 1/4 of the damaged endosomes had actually productive release. And even if there was release, the release was quite inefficient. So these are some of the inefficiencies then. So it's only a fraction of the LNPs that triggered membrane damages. And even if you have a trigger membrane damage, the release is quite inefficient. But it's actually worse. So in many cases, it can look like this, that we have an LNP here that triggered membrane damage, and then we have a certain release. However, in many other cases, it's like this. We have a complete void, and suddenly we have an appearance of a membrane damage. So if we pour LNPs on our cells, we have damages on LNP-containing vesicles, but we also have damages in structures that do not seem to contain any cargo. So that's yet another inefficiency. And especially with mRNA LNPs, only 20% of the damages that we see is on structures where we even have any RNA. So that's an obvious inefficiency that can contribute, of course, to toxicity. So then we ask ourselves, okay, might there be other mechanisms that is involved in the release? And one recently mechanism that seems to be involved in repairing damaged endosomes are the so-called escorts or chimp proteins. And if we pour LNPs on cells, we get spots with chimp recruitment to these LNPs. However, these smaller damages that result in recruitment of chimps do not seem to be associated with any actual release of the cargo. So we have recruitment of chimps, but no release. But that's still another potential inefficiency of the LNPs. So we have loads of membrane damages that are recognized by the chimps, but they are not associated with any actual release. And then using also other markers for different compartments in the cells, we can identify what structure inside the cell is it that is responsible for the release and the damage. And using such techniques, we can identify that early endosomes are defined by especially the RAB5 and EEA1 markers. There seems to be a window of opportunity for the cargo to get out of the endosome in those kind of vesicles. So then we ask ourselves, okay, what is it that is happening in these vesicles? Why do we see the escape in those early endosomes? So what is happening is that cells, they take up these lipid nanoparticles, they go into early endosomes, then they go into maturing endosomes, and then they're further sorted to late endosomes. And if we use super resolution microscopy, we can look at the early endosomes, and it almost looks like the cartoon here. So we can see LNPs inside individual vesicles, quite often tethered close to the endosomal membrane. However, if we instead look at late endosomes, then we can see that the LNP has disintegrated and dispersed the cargo inside the whole lumen of the vesicle. So what's happening seems to be that it gets in intact form and then it's dispersed. In the case of sRNAs, we have those on the left here. That's the case. However, with mRNA LNPs, we seem to get in early endosomes, it looks about the same. However, in late endosomes, down here, we see that the mRNA cargo remains aggregated, and it's not completely solubilized. So that might be an explanation why we can see a less efficient release with the mRNA cargos. Since we also see this phenomenon with a lot of damages on non-cargo containing vesicles, we thought perhaps it could be that the membrane active lipid in the LNPs, this MC3 lipid in our LNPs, might segregate from the cargo. And we addressed this question using a fluorescent analog of the MC3 lipid. And then what we see is this. So because of various photophysical characteristics, we can see when the MC3 lipid is in a dense structure, it has one color, and if it's in a free molecule, it's in another color. But so when we see it in a dense structure, we see it's present in the same structures as we have the RNA cargo. And we can see this also by looking at FRET, the proximity of the two molecules. However, looking at the free lipid of this membrane active lipid, we can see that it accumulates in perinuclear area in different structures than the RNA cargo. So they seem to segregate into different populations of vesicles. And looking with super resolution, we can see this also on the nanoscale. So here we have a single vesicle with the RNA cargo inside the lumen of the vesicle, while the membrane active lipid has jumped out of the lumen of the endosome into the limiting membrane of the endosome. And we can also see instances where we have a high concentration of the membrane active lipid in a certain region of the endosomal membrane. And actually, if we look also at the membrane damage marker, what we can see in many cases is that the semi-intact LNP is tethered on the endosomal membrane, and right at this very nano domain, we have also recruitment of membrane damage sensors, both this chimp and the galectin. So we seem to trigger a very localized damage inside the endosome. So what can we take home from this? Well, one thing is that the detailed understanding of the endosomal escape and the delivery process for most RNA delivery strategies at the moment is quite limited. So we are really just scratching on the surface of how this process proceeds and what we can do to improve it. What we know is that the LNPs, while functional, it's highly inefficient, and it triggers a lot of cellular machinery to protect the integrity of the cytosol. Small molecule drugs can be used to facilitate the release of compounds, especially for smaller RNAs. However, the balance between toxicity and efficiency is unclear, and it's also unclear what tissues you could use this. And LNPs are definitely needed at the moment for a larger cargo delivery, and also it's a question of what is the efficiency? Can we improve upon the LNPs better to deliver large structures like these super exons? And understanding this process is obviously very good if we want to improve our chances to take genetic therapies to CF, for example. That's all. These are the people involved and my collaborators.
Very nice talk. I wonder what you expect to be the consequences of actually disrupting the endosomal membranes, not just for the mRNA containing particle endosomes, but generally, because in addition to messenger RNA that we want to add, there are also physiological functions of the endosome.
Yes,
right?
Yeah, it's a very good question. So our belief, or some of our data, indicates that damaging later endosomal structures and lysosomes is not very good for you. It seems to be quite toxic. I would have thought that it would be even more toxic, but this cell seems to be able to handle some level of damage. However, damaging early endosomes seems to be much less toxic, especially with LNPs, for example, and also these lipophilic compounds I was showing. You can have really crazy amounts of endosomal damages. Of course, this is lab cells, but also, I mean, we give LNPs in the clinic, and it seems to be relatively tolerated. But I think, I mean, it needs to be studied more.
Yeah, and the second question is, can you actually target these early endosomes by, specific RABs, targeting the RABs, or?
So I think you need to target it by more physical-chemical strategies. So the molecules should have properties so you get the activity where you want.
Thank you.
Yeah, really interesting talk. I just wanted to ask a question related to the sort of observation of damage of vesicle endosomal structures in locations where you didn't appear to have LNPs. Can you talk further about then the, and I may have missed it, but is that escaped lipid? And do you have any thoughts about the sort of quantity of the endosome active components necessary to see that effect?
So how much membrane active component we need to get a productive release? Mm-hmm. Yeah, so that's a very, very good question, and something we are extremely interested in, and I don't have an answer.
Thank you.
So we have time for one more question right now. Afterwards, come back with the questions for the panel. Jeff.
Yeah, thanks. It was a really intriguing talk, and some of this may have been touched upon already, but the diffuse nature versus the RNA aggregates that you saw, it wasn't clear to me whether this is a function of the change in pH, the decreasing pH as the endosomes mature, whether it's new components that assemble with early versus late endosomes, or whether it actually has to do with perhaps just the sequestration that you're seeing. Do you have any sense of what the phenomenological basis of this is?
So there is a sequestration, I mean, or you get more cargo in later structures. That's one aspect, but it's also a consequence of both the cargo, so the smaller siRNAs, they disperse much more, and also the specific LNPs where they probably disintegrate because of the drop of pH. Yeah.
Okay.
Thank you so much, Anders.
So now we welcome the last speaker to the stage, which is Dr. Millie Sullivan, and is going to take us all the way to the nucleus.
All right. Good afternoon. Just wanted to, first of all, thank the organizers, Martin and Marianne, for the opportunity to talk today. And I'll try to borrow Anders' analogy that I think also represent a cat among ferrets, is it? So I'm also not a CF researcher, but really excited to share a little bit of what we are doing in terms of gene therapy and delivery to the nuclear compartment in particular. And so no conflicts to disclose. So just really quickly, I come from a chemical engineering background. Our lab is really interested in designing drug and gene delivery vehicles. And we do that really by thinking about the design on the one hand of the building block components, polymers, peptides, and proteins that will self-assemble in predictable ways to create a variety of different nanostructures and hydrogels. And then on the other hand, really interested in trying to unravel some of the structure-function relationships that are at play when those vehicles interact with cells and tissues during the delivery process. Most of our work is focused in the development of such approaches for regenerative medicine, especially wound healing of diabetic ulcers, and then also some work in targeted drug delivery for cancer therapeutics. And so I wanted to talk then today about our work, especially focused on nucleic acid delivery. I'll give you some quick motivations that come from the sort of engineering background, which include the interest we have in thinking about this delivery process as a really fascinating engineering problem of trying to figure out how to use materials to begin to shift the timescales of, on the one hand, processes like degradation and immune clearance that are going to take away delivery vehicles after they've been introduced into the human body. And on the other hand, this sort of set of biological filters and barriers and gateways we need to navigate, as we've heard nicely from some of the last speakers. And so we, again, are doing that by trying to control some of these physicochemical properties in the delivery vehicle using those self-assembly approaches and molecular design so that we can start to understand how those features are able to tweak the balance of those timescales and change lifetime localization and fate of cargo. There's also the fact that the timing is right. I think there's been this enormous growth in terms of the biopharmaceutical sort of portfolio of gene therapies. And so taking a quick look at that snapshot, we have this pathway from the 90s to 2017 with the first FDA-approved gene and RNA therapies to now in 2024, where we have 38 FDA-approved cell and gene therapies. And I have to update this slide every time I give a talk. And so this is a little bit of an apples to oranges comparison, but I think exciting point to be with this sort of growth in this type of therapeutic. But if we take, again, really messy math and say, well, 38 over the total number of gene therapy clinical trials, we're at a low success rate. And I think this continues to introduce this question of what we've been thinking about here, which is what makes DNA and RNA so difficult to use as medicines. And so we're also interested in thinking about delivery challenges in this process. And in particular, in comparing, for example, some of the pathways we have to do this, considering ways that will avoid some of the manufacturing challenges that are associated with alternative approaches for gene therapies. And so we've thought about these non-viral vehicles, as I mentioned, and how to navigate better some of the intracellular delivery barriers that exist. So advantages, as you heard, these are typically a one-pot assembly. They tend to be modular and versatile and not inherently immunogenic, but highlighting then that nuclear membrane as a particularly challenging barrier. We also know that there's a significant unmet or unaddressed block here in terms of how we can do better. And so my group has been interested in thinking about ways to take inspiration from natural delivery structures or natural structures that have features or capabilities that might be useful from an engineering context for creating better ways to get, for example, to the nucleus. In particular, we have started asking this question in the DNA delivery area about whether for DNA delivery, endosomal escape is always the most efficient way to reach the nucleus. And that's inspired by then the behaviors of a number of natural substances, proteins, viruses, and toxins that are known to evade lysosomes and recycling via retrograde transport processes, such as you see here schematically. And so we started in particular thinking about ways to try to create structures that might harness some of the components in things like histones, which, as you know, are the natural sort of packaging motifs used by all of our cells for chromosomal DNA. And so if we take a look here at the sort of cartoon version of chromatin, zooming in a little bit here on this nucleosome containing a histone octamer with DNA wrapped around, and then taking a look further at what is involved in this structure, what we'll see is that there are these components of each of the 8 histone proteins, these tail regions at the end terminus of each of the histone proteins that have really important roles in terms of dictating the delivery and sort of retention or redeposition of histones in the nucleus, as well as the delivery of those initial proteins to the nucleus. Also have a number of really interesting features in terms of activation of the unpackaging and transcription process, which are often dictated by the post-translational modification patterns on these tails. And so we've been especially interested in histone 3 tails for their roles in a number of aspects of these processes. So for example, H3 tails regulate DNA packaging and transcription. They are able to regulate retrograde transport to the nucleus. They're H3 effectors. Methyltransferase is known to do that. And they also regulate nuclear localization after synthesis and during mitosis via their interactions with importance. And so we took a simple formulation approach initially when we started this work of creating these nanostructures or polyplexes via self-assembly with plasmid DNA and histone 3 tails that we synthesized in the lab, followed by a post sort of complexation step with polyethyleneamine, commonly used for gene delivery, and started to ask just some basic questions about whether incorporation of these H3 tails would enhance gene expression, and if so, if we could pin down and uncover some of the mechanisms. Was this related to, for example, increased transport to the nucleus and/or activation within the nucleus? And are there any cell-specific effects? We also have looked at some of these sort of activation and unpackaging effects I'm happy to address offline, but not included in the talk. And so we were thinking about this work in the context of regenerative medicine applications and wound healing. So a lot of what I'll show you is on cells that may have nothing at all to do with cystic fibrosis necessarily, although I'll defer to others who know more about this, as we just heard, for sort of the cell type effects for CF. But when we looked at this in, for example, mouse fibroblast cells, what we saw is that there was an immediate clear benefit, at least in these simple models, for an induction of high levels of gene expression and a reduction in some of the cytotoxicity effects seen with these polymeric carriers. We took this further to start to, again, try to understand the navigational process that these vehicles were using. So, for example, hypothesizing that while PEI polyplexes have been diagnosed to use these sort of endolysosomal escape pathways that we heard about in a different context from Anders, we predicted that potentially the inclusion of this H3 peptide sequence would shift toward some of those H3 specific pathways. So for example, a retrograde transport pathway through the Golgi apparatus in the ER. And so we also looked at this by utilizing some of those components that allow us to take a look at what is happening after we deliver these polyplexes. And so we use these RAB GFP fusions to begin to take a look at where our polyplexes were going. And we did that in essentially a pulse chase experiment of delivering for a short time period labeled polyplexes and then following them through this set of compartments as a function of time and live cells. And so here you see some of the evidence that we collected for the H3 targeted polyplexes. And here in particular, what you're looking at is sort of a time-dependent set of transport steps through early endosomes, late endosomes that transport towards the trans-Golgi network, recycling endosomes, and these sort of vesicles that shuttle between the Golgi apparatus and the ER. And so what we saw is evidence of, on the one hand, transport through endolysosomal and/or recycling pathways. And so we can hypothesize that potentially there's sort of a handoff process happening here from early to late endosomes, and in some cases, recycling endosomes. But we also saw what looked like evidence of these retrograde pathways that we had theorized might actually be activated by H3 inclusion. So for example, we saw transit through RAB9 labeled late endosomes that shuttle towards the trans-Golgi network. We also saw, interestingly, an accumulation, as what it looked like over at least this initial 4-hour period in RAB6 labeled vesicles, which we thought was interesting. And so we started to look further at this and also to compare with the non-peptide targeted structures. I'm here tracking co-localization as a function of time, and these are in synchronized cells so that they would divide all at the same time. But co-localization with RAB6 as well as nuclear markers. And interestingly, this accumulation in RAB6 was an effect that was observed only in the H3 targeted structures, suggesting that that was a more specific effect to this peptide as predicted. Additionally, when we take a look at the sort of transit process through these RAB6 vesicles where we hang out for a long period of time, there was a correlation between when Rab6 colocalization went away and when nuclear colocalization increased for those H3 labeled structures. And so this led us to ask if there was some alternative mechanism by which these H3 structures might be accessing the nucleus, because it did not necessarily appear that they were going through an endosomal escape, and rather we started to think about whether the ER membranes, which are known to be a source of new nuclear membrane after mitosis, might somehow be responsible for, or binding to those membranes might somehow be responsible for our H3 structures to make it into the nucleus. And so we've looked for evidence of this type of a phenomenon in a couple of ways, and I'll just show you a few of the sort of pieces of data that we have. So one was to use super resolution microscopy and to take a look, again in synchronized cells, at the size distributions of these clusters of our labeled polyplexes as they go through the cell, hang around in the sort of perinuclear compartments, and then the cells go through mitosis. And we saw what looked like a sort of a interesting shift, in particular where these sort of clustered structures were the ones that appeared to be, or vesicle-sized clusters, were the ones that were landing in the nucleus. We've also used fluorescence correlation spectroscopy as an alternative tool to take a look at this. And so here, in particular, using this tool to get a sense within live cells of the diffusional timescales of the fluorescently labeled sort of signatures or structures that we see. And so here in particular, also seeing evidence that corresponded with the microscopic evidence, which suggested that we had a set of sort of slower diffusing and more vesicle-like structures in the cytoplasm that then shifted and flipped over into the nuclear compartment following mitosis. So again, some interesting evidence indicating that we really have a larger sort of clustered set of stuff that's going into the nucleus as opposed to something that is free within the cytosol. We further started to take a look at the impacts of this H3 peptide inclusion on what happens inside of the nucleus. And so again, also using microscopy and colocalization analyses here with now red labeled polyplexes and colocalization with chromatin. And we started to see some evidence of a nice level of colocalization. We also started to probe mechanisms of what might be enabling and driving this effect. And we did that by taking a look at important 4 and its importance in mediating this effect. And here, if we use an siRNA inhibition of important 4, what you see is that that abrogates the colocalization effect as well as most of the nuclear delivery, indicating that important 4 is likely important for both the delivery into the nucleus and potentially mediating these chromatin interactions. And so here, that quantified data here, just to give you a sense for what that looks like. And so what we have been thinking about sort of moving forward, and I just put in a couple of tidbits here, but happy to answer questions about the longer underlying story, but we theorize that while unexpected that H3 would have a sort of a preference for these retrograde pathways, or at least we were expecting some different effects, but the caveolar trafficking preference and this retrograde trafficking preference we saw for these H3 structures led us to think that potentially there might be some cell-specific effect in terms of our ability to deliver gene therapies. And so this is something here which I've just given you a snapshot of, but we've taken a look at this for example, applications related to breast cancer-specific gene therapies, and in this case, showing through a simple co-culture model of inflammatory breast cancer cells which heavily overexpress caveolin-1. We co-cultured those with breast epithelial cells and are able to show that in that co-culture model, we get essentially equal gene expression when using the non-targeted structures. If we now flip over and start using those H3 targeted structures, you'll see that there's a high level of gene expression just in the inflammatory breast cancer cells, which then we think, based on some of the additional work we did here, is related to caveolin-1. So if we silence caveolin-1, then this effect goes away. And so this is some unpublished data I just thought I would share because it goes, and in our case, it's a different application from CF, but we've been interested in collaboration with physician, Deepthi Alapati at Nemours Hospital, and beginning to develop gene therapy approaches for infant lung diseases such as bronchopulmonary dysplasia. And so for that, we've been starting to take a look at formulations that might have properties that are relevant to efficient gene expression in such cells. We've started to create a hybrid formulation using lipid nanoparticle components and these H3 peptides, which have so far shown some interesting but really sort of very preliminary results. And so here we do sort of an initial DNA peptide complexation, and then we're doing a post formulation with LNP components. We're using some of the sort of standard ratios that you've seen in terms of the lipid formulations based on what we know has been used already in a number of applications for vaccination, et cetera. And interestingly, and this is something we've seen in mouse lung epithelial cells as well as in human lung epithelial cells, we're seeing that these H3 and lipid components together appear to have a really synergistic effect. However, we don't really know quite what's going on yet, and we haven't done the work to figure it out. We're seeing a scrambled peptide has a nearly similar effect. And so it's not clear that this is necessarily based on that sort of specificity or H3 particular activity that we've seen previously, but I think an interesting effect that we're currently digging into. And so just to summarize, we've been excited to be working on these peptide-based approaches to try to sort of retarget and reshuffle and redirect nucleic acid cargoes within cells. In particular, these histone structures appear to have beneficial effects in terms of enhancing gene expression, particularly in caveolin-expressing cells. And in doing that by harnessing components of the native nuclear localization machinery, we've actually used that for delivery of growth factors, again, for different applications that I didn't show you today. But excited to sort of explore further for additional target spaces as mentioned. And so just some quick acknowledgements. In particular, I'll cite a few students, Eric Munsell, Nikki Ross, and Hugo Hu for their contributions to the data collection in this work, our funding sources, and I'll leave you with a quick picture of my group and happy to take questions.
Kevin Foskett up the road at Penn. Nice to meet you.
Nice to meet you.
Have you up. So I'm a little unclear about what you think the mechanism is. On the one hand, you're talking about retrograde transport. On the other hand, you said mitosis, where the nuclear envelope would break down, was important. And then you also said important, which would be a transport-based mechanism. So 3 different mechanisms. So are they all working, or what do you think?
So we have some additional, I didn't show all of the pieces of this, but I'll try to navigate what we think is happening because it's a little, so we think there's an endocytic step that is then the sort of initial, I don't know that we have a great explanation for why histone peptides would induce retrograde transport, but we've seen that. We had been scratching our heads about how something in the lumen of an endosome would then bind to something like important 4, for example, which is on the other side of the membrane. We have some evidence, and it's published, and I probably should have put that in, but some evidence published in some of our papers that shows that there is an interaction with one of the sort of transporters that can flip cargoes to the other side, and so that's a possibility. And so we have a number of pieces of circumstantial evidence that are showing involvement of different molecular components, and so we have this partially constructed theory of, okay, so there's a retrograde transport step that is involving histones, but why specifically histones for that, I don't know. I think there's a flipping process that's allowing access to importance. And so I think that is where that comes into play. I think the polyplexes are too big to use sort of a nuclear pore assisted process to get into the nucleus, which is what we've seen in literature for a number of these structures. And so we do see that these same processes of access to the nucleus, they don't happen for all structures. I don't think there's sort of a blind sorting into the nucleus that happens. And so having a nuclear localization sequence seems to be important even during mitosis. So.
Hi, I'm Hilary Valley from the CFF lab. Really fascinating talk, thank you. I was just curious if you had done any work to like shorten the tails you were putting in from either direction or really understand what are the essential components, and was wondering if that could help inform mechanism. I'm not sure how much of the tail or if it's the whole tail that you're putting in. You mentioned a particular transferase, so like potentially that particular part is important, but have you tried shortening it? Like, does it still work if it's shorter? Are there any insights there?
Yeah, so it's a good question. So what we're using is the 25, starting at the N-terminus, the first 25 amino acids of the H3 tail. We've not tried shortening that to see if there's like what is the essential component or piece of that. So I don't know. We selected that sequence based on what was known about the sort of the nuclear localization region of the histone protein overall, which is somewhere in there. But exactly which region, I don't know. So yeah. Thanks.
And a nice presentation. I was curious about your strategy of using fluorescence correlation spectroscopy. Were you able to distinguish between cargoes that were free in the cytosol versus the nucleus, or was it if the cargo was still in the complex or outside of the complex?
So the question is, with FCS, can we distinguish structures that are free in the
cytosol versus the nucleus, because you would assume you have a less diffusion within the nucleus?
Yeah, so you can with FCS. I mean, your sort of your focal region, if I remember correctly, is like a one, and I'm sure at least the scope we were using is like a 1 micron sort of square cubical region. So you can focus within a particular compartment of the cell.
With your experiments, did you distinguish between the cargo being inside the complex or?
Yeah. So we can't, based on what we did. I think we would need labels, fluorescent labels on both components, the DNA as well as the carrier. We've seen that labeling the peptide, which we've tried to do with fluorophores, changes its binding significantly, and so then we get a completely different behavior overall because the peptides ultimately aren't that big compared to the labels. So we haven't been able to pin down those questions of who's going where, which would be interesting. So thanks.
So while Margarita is asking her question, I just invite the other speakers back up so we have the full panel up here. But go ahead, Margarita.
Thanks. Very, very interesting. So instead of putting the histone tail, have you ever thought about just introducing NLS, nuclear localization signals?
We have not tried. I think a lot of the motivation for this work was other behaviors of the H3 tail. So we were looking at trying to combine the nuclear localization piece, which initially I wasn't sure would work because these are big polyplexes. And so I'm glad that it did seem to have an effect. But actually we started with H3 because we were interested in the sort of chromatin and transcriptional activation process. We've also tested, for example, post-translationally modified H3 tails to look at transcriptional activation. So we haven't tested other NLSs because we had that sort of dual interest. So it's an interesting question, but I don't know the answer to what would happen there. So.
And regarding the tails, are they acetylated?
Are they acetylated? No. Yeah.
Okay, that might be interesting as well.
We did a trimethylated tail, so we've compared just a non-modified tail within the it's the fourth lysine, so H3K4 trimethyl group, and so that seems to accelerate transcription. So we showed that by using some nuclear microinjection experiments. So we have tried post-translational modifications, not acetylation, but methylation, and it does seem to affect the transcription process. So.
Okay, because methylation is usually associated with silencing and acetylation with enhanced.
Yeah, so H3K4 trimethyl is actually heavily associated with activation and with expression. So it's got a series of effectors that bind to it to recruit transcriptional machinery, and it's associated with loose chromatin as well. So.
Okay.
So I invite Millie also to sit down, and then we can continue the discussion with questions for all the speakers. So Devesh.
Hi, Devesh from the CFFT lab here. Actually, another question for Dr. Sullivan. So I was wondering if you investigated into like other types of histones and maybe they have different characteristics or like maybe different target preferences or something of like that.
Yeah, it's a good question. We did a little bit, not published, and it's been a while. So we've tried a little bit sort of combining H3 tails with other tails within the histone structure. We didn't see significant additional or additive effects, and so we didn't take it very far. But to be fair, we also just probed a little bit of the space, so I think it's an interesting question.
Yeah, this is Tom Harris from UAB. I have a question, a comparative pathology question for Dr. Maherholtz. There's a question in your talk about the distal airway being a seed point of CF. And I wanted to follow up on that as far as, do you have comments on which animal models might be the best for studying the distal airway, whether anatomically or cell composition? And then probably just as importantly, which animal models really don't replicate the distal airway? And I guess that kind of folding into, are animal models satisfactory to that, or are we still stuck basically studying human tissue?
I assume you're wanting to throw tomatoes at me. I think each animal model does have advantages and disadvantages, and I guess it really depends on what question you're asking. And so ultimately, if you're talking about the origins of the CF disease, I'm not even sure we fully understand that yet to make it a definitive statement. But as far as for studies, those who get a spontaneous disease or induced disease, we can study from those and learn them together. So I don't want to say there's a best one out there, so to speak, but really we can learn from all of them together.
Anne Harris, Case Western. I've got a question for Dr. Gompertz. That was a beautiful talk. It's a question that's kind of broader. It's not really about submucosal glands, but you defined your cycling basal cells, which I assume are cycling based on centromeric markers, DNA replication, etc. And when you look at larger portions of lung tissue from your previous data, are all the cycling cells that you see then, are they all basal cells, or do you think there are other cell types, other identities that are also dividing fast and therefore they form a larger cluster?
Yeah, it's a great question. It was actually something that came up at the poster session earlier. So the answer is I don't really know. I think we need to go back and look at our data, is what my conclusion from the poster session anyway. The most obvious ones are the basal cells that are cycling. Could there be some serous progenitors in there that are also cycling? I think it's very possible. I also think neonate, sort of neonatal tissue versus this is adult tissue may be different. So I think we need to sort of think not just about one snapshot, one point in time. Also injuries, right? So with different injuries, you may turn on different, progenitors in terms of needing to repair. And I think this comes back to Tom's question earlier about the mouse. Maybe in the setting of a particular injury where you're sloughing off your entire epithelium, maybe you're going to be inducing programs that are going to be similar to what we're seeing in the human. So I think those are great ideas and things we should look deeper into. Thanks.
This is Wenjie from University of Iowa. I have a question for Dr. Gompertz. I'm wondering, so you show different growth factors stimulate gland organoid proliferation or formation or differentiation. I'm wondering, you saw some data like non-epithelial cells secrete those stuff. I'm wondering what specific cell type produce those like growth factors, and potentially, have you ever looked at like immune cells, their role involved in gland enlargement or growth?
So the question, if I'm getting it correctly, is that you're wondering with our different kinds of submucosal glands, like the duct progenitors versus the myepithelial cell progenitors, if there's an immune component in that? Was that the question?
Yes.
So that's a wonderful question, and I think to study that we'll have to add like macrophages or other immune cells into the actual organoids to really be able to, in real time, look at those interactions. I think that anytime we look at like serous secretions in particular, we get this beautiful immune signature. And so that is definitely there. We even have sort of an immune cilia population that we described before that we see again. And so I think innately and inherently, yes, the glands do have an immune signature. And that I think could be driven further if we added in, some of the immune compartment.
Thank you.
So before I go back to the audience, I would like to actually ask a question of Anders Wittrup, and maybe Millie has something to say about that too, but I was curious. So endosomal escape, as Anders beautifully illustrated, is a big bottleneck. And there's also strategies people are working on just bypassing the endosome altogether with fusogenic peptides, cell-penetrating peptides. From your perspective, is there a potential benefit to actually go through the endosome? Does it serve as a meaningful filter? Could it be sort of extending actually exposure like the pharmacokinetics because it's a slow release depot? Any thoughts on that, if there's benefit to going through the endosomal pathway?
Yeah, I think you have many good points there. I think so. So you can use the endosomes as a depot for obviously for siRNAs. It has been shown now that you can have activity for half a year or even a year. So but I think that might be a more a specific case when you can have really good internalization, and then you can accept very bad or low degree of endosomal escape, but over a long time. I think going through, another advantage of going through the endosome, I think, is otherwise you would have to have some sort of membrane damaging activity that would be active while it's still outside of your target cell. So then you would have a risk of having toxicity or activity on even in tissues which you actually do not intend to target. And then secondly, I think it seems a little bit easier to have some sort of trigger, and then you can have an activity. A lot of viruses go through the endosomal system. Obviously, not really all, probably. But I haven't seen fantastic data showing fusion at the cell surface with a non-viral delivery vehicle. It might happen, but I haven't really seen the data.
Okay, Emily, I assume if you want to use your mechanism like the histone peptides, you're also better coming from the endosome rather than sort of delivering to the cytosol.
Yeah, I was thinking about the answer to your question for maybe alternative approaches as well. I mean, I think from a more general perspective of developing stimulus responsive chemistries and utilizing those, and maybe this is kind of building on Anders' last points, but I think from that context that there are a number of ways to create materials that will, for example, not just rupture endosome, but also release cargoes activated by pH. And so I think a benefit of the endosome is also that you can activate both a release of cargo and also endosomal escape potentially in one fell swoop.
Well, thank you.
Hi, I have two questions for Anders. One of them is I'm curious about the level of endosomal escape with LNPs as opposed to just what's normally happening in the cell.
So I think you massively increase it. It's not zero, as evidenced by, for example, the GalNAc targeting siRNAs without any membrane active lipids or similar things. But I think, you increase it hundreds or thousandfold. It's a very low. We've obviously looked at the galectin recruitment spontaneously in hope of catching the mechanism of GalNAc siRNAs, for example, but the activity is really low. It might be more for the smaller damages like the chimps.
Do you know what is inducing that level of escape?
The natural level,
or I guess either natural or the artificial.
So with the LNPs, you trigger it. It's especially the ionizable lipid. So the one I referred to as MC3 in the ones we used, but the other ones are using other. Those have properties that disrupt the membrane. So that's the trigger.
And then how specific is your galectin-9? Like, do you know it's the exclusive labeler or
no,
one of them?
It's not 100% specific for membrane endosomal damages because we see spots in the nuclei of cell which are really annoying for automatic identification of events. But we have no idea what those are. it's doing something.
All right, thank you so much.
Grab a question here.
We're going in order.
Can I quickly ask one question for Anders because I was on the same topic? So you've talked a lot about the efficient release of relatively efficient release of siRNA, but it seemed a lot less efficient for mRNA. And then I wonder, the whole concept of LNP mRNA delivery is that theoretically you can put very large mRNAs in it. Is your feeling that the efficiency will drop down even a lot more? And should we be concerned about that or not?
So the LNP we've studied the most is MC3 based LNPs, and those are not, they are okay for mRNA delivery, but they are probably not the best. So it would of course be very interesting to look at more mRNA efficient LNPs. I don't think we should abandon that pathway.
Okay.
Oh, hello. My name is Hamide. I'm from Cincinnati Children's. So my question is about anatomy. I think my question is for David. So you mentioned we have some challenges for physical delivery of the drug particles to the target site. So my question is, has there been any study on that part? Have you done any studies to see where those drug particles or gene therapy particles, where do they get stuck? Where do they trap those particles?
From an intravascular standpoint, or
gene therapy particles?
Right. We've done some studies in localization and trying to get delivery of gene therapies to the lung and where they go and that sort of thing, and those have been published. But as far as other ways or other mechanisms other than the topical aerosol therapy, less so. There's not as many studies out there that we've done, at least.
Oh, okay. Thank you.
So I think that will be our last question coming up. We are at time and everybody wants to go to the plenary, I assume, but Kate, please.
Thank you. So Kate Coffin from Spirevant, and this has been a great session. So kudos to the organizers and to all of you in thinking to put this interesting path together for everything. But I have a question for Millie. There's circulating DNA naturally in the body, and I don't think it's really well understood. Does that have histones, and do you think you've tapped into a natural pathway that may be doing more than anybody understands or knows?
Yeah, I'm not sure. We've done certainly some sort of scouring of the literature related to extracellular histones. So I don't know that that's clearly the case, although it's an interesting hypothesis. I think we landed on, and I welcome further suggestions or discussion. I think we haven't landed on a molecular target to sort of test hypotheses associated with that. And so I don't know, but it is something we've thought about, just haven't gotten particularly far on testing anything substantive. So yeah, it's a good suggestion.
Well, I thank the audience and in particular the speakers one more time here for really an excellent session. So.